Okay, so I'm here today to introduce Alyson.
Hey Katie!
Hi Alyson!
How's it going?
Good!
Am I late for JClub?
No, you're good. I'm just about to start introducing you.
God.
Okay, ready?
Ready?
Okay, so I figured I'd start by talking about some of the good times we've already had together. So I was starting out by thinking about the time we tried to have fluff in our sandwiches, but you seemed to struggle a little bit.
Oh dear, that's so embarrassing.
The time we tried to have lobster, but that also didn't quite work out so well for us.
He's so cute.
Okay, I know, it's only
You see how I let it go?
So that kind of got me thinking about what was Alyson like before she came to Boston. So I thought, she must have had these really cool friends, really smart, just great friends, and then, saw that.
Oh my god, that's so crazy.
And then I was thinking about, okay, so what was Alyson's, previous boyfriends like? It must have been really, really nice, attractive, right? Then I kind of found that, so. Yeah, but that's a bird, Sean. You're not the only one.
These stories are really embarrassing, Katie. I hope you didn't put up that one time I got jizzed on by a giant.
Oh, like this? Yeah, but despite this all, we know that Alyson's gonna be really good today. She's always ready, super prepared for life. So good luck, go Alyson!
Thanks for that introduction. It really pointed out the finer parts of my personality. So I'm Alyson. I'm the first G1 to go for Journal Club, so I thought that I would give you guys a nice snapshot of our class before I begin. So if we look at this graph right here, we're looking at the average G1 time of arrival to our 10 a.m. class. And as I was making this graph, I noticed that there's particular students who follow along this distribution consistently every single day. But you've been doing better, so we'll put you up here. So we have our students who consistently come late, and then we have our students who consistently come early and prepared and really peppy and excited for class every single day. So I think that my fellow students are phenomenal scientists, so I was thinking to myself, maybe there's some sort of biological reason underpinning this. Maybe these students can't help the fact that they're late every day. So that took me to some sleep-inducing research and our biological clock. So as Javier introduced us last week, our biological clock is entrained through a number of different parts of our brain. We have light coming in through our eyes, which projects to our retinal ganglion cells, which projects to this red box here called the suprachiasmatic nucleus. And this in turn will regulate the rhythms of our neurons throughout our body, and in turn, we need some sort of hormone in order to regulate all these different biological clocks throughout our entire body and our brain, and essentially tell us when it's night or daytime. And this hormone is called melatonin. And so the pineal gland right here receives information from the suprachiasmatic nucleus, and it will secrete melatonin, which will project to all our different body areas and keep our biological clock on time. So the upregulation of melatonin at night has led to its characterization as the hormone of darkness. And so if we look throughout evolutionary history, humans aren't the only ones who have melatonin. We can see that melatonin is found in a bunch of these different taxa throughout evolutionary time, and we can see that its cyclical expression is actually found in a number of different taxa. So in bacteria, we see that it's upregulated at night, and we don't see its synthesis during the day. In plants and animals, we also see cycling melatonin expression, and most interestingly, we see melatonin in both vertebrate and invertebrate species. But melatonin is not always associated with this sleep-related behavior. In fact, if we look at these more simple organisms, we can see that melatonin first came about as an antioxidant for getting rid of reactive oxygenated species within these different taxa. So an interesting question to ask is if we find evidence of a circadian role of melatonin deep within evolutionary history, maybe we can first identify when melatonin first became a hormone of darkness and became associated with these behaviors that we see only occur during the nighttime. So that brings us to this simple invertebrate species, Platyneris dumerilii. which is a marine annelid worm. And if you're not great at drawing, this is what the adult species look like. But most interesting about this worm is that it has a marine zooplankton larval stage. So if we look at this larva right here, in red I've drawn the episphere, which is our larval brain. And all around the larva, kind of like a tutu, there's a cilia band, which we call the prototroch. And the cilia band will move to control the locomotion of this larva within the water column. So if we look a little closer, we can see that there's actually neurons within the larval brain that project these cilia and help them move. And these invertebrates show this really, really interesting cyclical migration that we call diel vertical migration. And so if we look at the height of my paper as the height in the water column, and the length of my paper as the time during day, Platyneris dumerillii larvae will actually change their height in the water column as a function of what time of day or night it is. So these larvae will reach their highest point in the water column at dusk, will fall through the water column at night, and reach their lowest point at dawn. And so what's interesting about this is that vertical migration is, if we add up the vertical migration of all of these plankton that are found within the ocean, it actually accounts for the largest migration in terms of biomass in the entire Earth.
How much do they travel?
I think it depends upon the species. So this one is found mostly in shallower underwater environments, so I think it's only a few feet. So the purpose for this has been postulated to be a number of different things. The first could be escaping predators. So if we're moving through the water column, we're escaping predators that prefer these warm surface waters or lower waters. It could also be a metabolic advantage or for energy conservation. So if we're found up here at the surface waters and we're eating all of our food and digesting it in the nice warm waters, when we go down to these colder surface waters, we don't have to worry about digesting our food when it's cold. And it also could evolve to avoid UV damage. And this is where melatonin really comes in. So if we're avoiding all of these UV rays that come in during the day by being in these lower waters during the day, we can see that we're avoiding a lot of the sunlight that comes in through the ocean and could potentially cause a lot of these reactive oxygenated species, which could cause damage to these larvae. So that brings up the following point. We've identified a circadian pattern of behavior in invertebrate, but these invertebrates need some sort of way or a signaling molecule to tell if it's night or day. And that brings us to the hormone of darkness, melatonin, and the paper I'll be talking about today, which is melatonin signaling controls circadian swimming behavior in marine zooplankton. This paper came out in Cell about 2 months ago. So the first thing that we need to establish in these invertebrates is that we actually have some sort of clock-like behavior and that this crazy migration is due to an entrained circadian clock. So the authors performed a qPCR screen in which they looked at 2 different sets of genes. They looked at a whole bunch of circadian or clock-related genes, including those involved in melatonin synthesis, and a whole bunch of developmentally regulated genes. And they found the following. So in blue here I've shown a low expression, and in red here I'm showing high expression. And if we look at the expression pattern of these circadian or clock-related genes, we can see that they have low expression during the day, goes to high expression at night, and again falls to low expression during the day. Conversely, our developmentally regulated genes follow a linear fashion just based on the age of the larva. So we have low expression during the day, or low expression at 45 hours post-fertilization, which increases as the larvae age. And if we parse these out into individual genes, we actually see the same cyclic behavior. So here on our x-axis, sorry guys, we have our times of day and night. And we can see that if we look at the expression of particular genes, that again, the way these genes are cycling falls into a couple different categories. So first we have our clock genes, which are low during the day, cycle to high at night, and go back to low. Our developmental genes follow pretty much a linear fashion. There's this whole other group of genes that don't really fall into either one of these. And so the authors indicate that there's a whole group of these genes that they're looking at, but they don't really go any further into explaining them, so we'll leave that alone for right now. So what happens if this light cycle is inverted? So in order to establish that we actually have a circadian clock within these plankton, we need to show that the cycling of genes is not just entrained into the first cycle that they see when they first are born. So if we compare the expression of these genes in 2 different sets of larvae, the first is our wild type, where again we go from day to night to day, and compare them to light inverted larvae, we actually see that these genes still retain this cycling pattern, in which we have high expression during the night, low expression during the day, and high again during the night in our light inverted larvae here. And conversely, there's other genes that cycle in the opposite manner, where if we look at our light inverted larvae, they're low during night, high during day, and we can see the opposite in our wild type. So we've essentially established that there's some sort of circadian clock. So since we've established that there's a circadian clock, we also need to see how melatonin plays a role in this process. So the authors noted that there was two different behavioral states that they saw within these larvae in their cilia. The first is this open state in which our cilia are moving, so our tutu is activated essentially. And these plankton have a net upward movement through the water column because they're pushing themselves upward. We also have a closed state in which our cilia are lying down at the sides of the larva, and this results in a net downward movement because these larvae are not actively keeping their place in the water column. And so the authors performed the following assay, in which we hatch our larvae at the exact same time, put half of them into a night cycle and half of them into a day cycle, let them grow up a little bit past 48 hours post-fertilization, and perform an assay in which we're seeing how these cilia are acting, if they're staying in the open or closed state, or like what their preferential state is. And so if we calculate the percentage of time that these cilia are spending closed in both the day and night state, and the number of closures per minute, we can actually see that there's a significant increase in the night state in the time that these cilia are spending closed. And so that's what we would expect for ideal vertical migration. If our cilia are closed, we're going to have a net downward movement through the water column. So what role does melatonin play in this process? If we add in melatonin to our day larva, we can see that in our day larva with melatonin, we have an increase in the percentage of time that these cilia are spending closed. And conversely, if we add a melatonin receptor antagonist to these night larvae, we can see that there's a decrease in the percentage of time that these cilia are spending closed. But this is not the entire story. So if we go back to our behavioral assay, there's actually two different states that we see for these closed cilia. The first is this closed one, which is we characterize as short, and it lasts for only about 0.2 seconds. And the second is this long closed state, in which we lasts for about 2.2 seconds. And the distribution of these two falls into a bimodal distribution, in which we have a high number of these short closures and a lower number of these longer closures. And again, if we characterize the percentage of long closures, we can see that there's an increase in the long closures in the night state. And so if we look at the distributions of both of these night and day larvae, we can see that the percentage of closure length or the number of closures per minute in our night larvae increases in both short and long closures as compared to the day. So what role does melatonin play with this? If we add a melatonin receptor antagonist to our night larvae, we can see that we completely ablate these long closures, and we significantly reduce the number of these short closures, indicating that the addition of melatonin is sort of creating what we see in our day state. If we add melatonin to these day larvae, we can see that we're almost completely recapitulating what we see in our night control. So we see an increase in both these long and short closures, indicating that melatonin plays a role in regulating how these cilia are beating. So what next? In humans, melatonin signaling dampens neuronal firing within all of our peripheral organs, which promotes a sleep-like state. So if we look at our suprachiasmatic nucleus again, it will activate this pineal gland, which secretes melatonin, which projects to all of these other organs. So humans are a lot more complicated than the invertebrate that we're looking at, and we want to see something a little bit similar. So how about dinoflagellates? Dinoflagellates are a single-celled organism that exhibits dual vertical migration, expresses melatonin, but has no circadian clock. So the authors want to prove that this link between melatonin and behavior is kind of halfway between this. So we're seeing something that's more complicated than dinoflagellates, but obviously less complicated than humans. So I think a fair question to ask is if the neurons that drive our prototrophs or cilia behavior sensitive to the hormone of darkness or melatonin. And so to establish this direct link, the authors did 2-photon calcium imaging to see how the neurons within the episphere change according to melatonin signaling. And so if we look at our day state, we can see a very short baseline of firing. But if we look at our night state, we can see the change in fluorescence creates this rhythmic behavior. And so what role does melatonin play in this process? If we add melatonin to our day state, we can see that we immediately go from baseline firing to this rhythmic state that we see at night. If we add a melatonin receptor antagonist to our night state, we can see that we immediately recapitulate the change in fluorescence that we see during the day. And if we add to our day state first melatonin and then a melatonin receptor antagonist, we see that we again recapitulate this day state. So this is directly proving that melatonin plays some sort of role in regulating how these neurons are firing. So these neurons project to the cilia. So how do the cilia feel about this? To test this, the authors performed the following experiment. So the neurons that are involved in the larval episphere that project to these cilia release the neurotransmitter acetylcholine, which is an inhibitory neurotransmitter. So we expect that when these neurons fire, our cilia will arrest. And so they did intracellular recording within these cilia to see the membrane potential and image the cilia at the same time to see if they're arresting at the same time that we see our membrane potential changing. And so if we look at the day state, we can see that these cilia are depolarizing at a very sparse rate. And we also see that at the same time, our cilia are closing whenever these neurons fire. If we add melatonin to this day state, we see a rhythmic process in which these neurons are firing at a much faster bursting rate, and so our cilia are also arresting or closing sooner. And so if you look at the excitatory postsynaptic potential recording, so this is recording the information that these cilia are getting from these neurons, we can see during the day state we have sparse firing, addition of melatonin to the day state and again induces rhythmic bursting, addition of melatonin and a melatonin receptor antagonist to the day state again recapitulates this sparse firing. And if we add an acetylcholinesterase inhibitor, we're completely wiping out endogenous function, which is showing that these neurons do project to these cilia, and they are sort of regulating how they move. So based on the evidence that they presented, we can come up with the following checklist. Is melatonin signaling in plating neurostimuli linked to this circadian rhythm behavior? We've shown that there's a circadian clock. We've shown that a nocturnal release of melatonin affects behavior. And we've also shown that the neurons that drive prototroch behavior and the cilia themselves are melatonin sensitive. So from this checklist, the authors came up with the following model. During the daytime, we have no melatonin synthesis, so these neurons are free to fire whenever they want. They fire sparsely, which leads to less ciliary arrests. At nighttime, we have increased melatonin synthesis, which induces a rhythmic bursting of firing within these neurons, and we have more ciliary arrests. So if we parse out what this looks like within the cilia, we can see that the cilia during the daytime have the freedom to stay in this open state, and so we have a net upward movement. Whereas at nighttime, these cilia will remain mostly in the downward state or closed state, so we'll have a net downward movement. So the authors came up with the following conclusions. In Platynarius, melatonin is necessary and sufficient to establish a nocturnal behavioral state characterized by enhanced ciliary arrest. So they parsed this out into two different components, the first being the sensory modulatory component and the second being this effector component. So for the first, we've established that we have a circadian clock and that there are photoreceptors within these larvae that respond to light. Secondly, we've established that the ciliomotor neurons are direct targets of nocturnal melatonin signaling. And from all the evidence that they've shown in this paper, the authors come up with the following conclusion. That this system represents a minimalist example of integration between sensory, neuromodulatory, and motor circuits and may be evolutionary ancient. So I think that this is a little bit overstated. The authors have shown some really convincing research that melatonin is first linked to this nocturnal behavior, which has never been shown before in an invertebrate. But instead they go on this whole thing about how what they've demonstrated is this evolutionary, like the first evolutionary link that we see, and is the context in which this whole melatonin linked to nocturnal behavior concept evolved. So I still have a few remaining questions. The first being that a marine zooplankton does not equal all zooplankton. So they only tested one zooplankton that exhibited this behavior, but again, zooplankton is sort of a way of life. It's not necessarily a categorization for a single type of organism. So I would like to see this in a few other zooplankton before we can necessarily say that this is something that we see within this group of organisms. Secondly, I want to know how close to a sleep-like state is deal vertical migration. Does a differential arousal state exist, and if so, is it regulated by melatonin? And lastly, I'd like to assess how changing melatonin levels affects the actual height in the water column. So the authors don't ever actually put these larvae into a column and see how melatonin signaling affects their height within the water column. And that's something that I'd really like to see. So with that, I'd like to thank my coaches and open the floor for questions.
Do you know off the top of your head some of the genes that they use as examples?
Yeah, so for their circadian genes, they use alpha and clock and HIOMT, which is one of the enzymes involved in melatonin synthesis. And one of the ones that they couldn't categorize is pyropsin, which is an opsin-based gene, which is kind of interesting to me that they couldn't really categorize it. And for the developmentally regulated genes, they use SIRT, which I think is some sort of receptor protein. I'm not necessarily sure.